is Ho Yu. I'm at Columbia University, and I'm going to follow on from what Alex was speaking about in terms of some of the research that's going on in terms of the three diseases that we're focused on today and through CurePSP, that's PSP, CBD, and MSA. And it's true that Alex has kind of discussed some of the plans that go on and some of the importance of the connections between neurons with each other, as well as how glia can help support these things. And it's really important to kind of understand that and remember that neurons have basically run everything. And it's not just memory, it's also movement, it's also the fundamental things that one doesn't even think about that the brain went to, including regulating your heart, regulating your lungs, everything. And it's a fine-oiled machine most of the time. And that's really what we want to get to, is kind of keep that efficiency going in terms of the brain. That's not just neurons that are involved, but also making sure the glia are part of this whole process. And what we understand from PSP, CBD, and MSA really is an evolution of the discoveries, not from just these diseases, but what we can learn about brain function itself, as well as other diseases like Alzheimer's and Parkinson's disease, which have a lot of funding. And throughout my talk, you'll see that there are commonalities with diseases. And so what we learn from other diseases, we can also play into this. And similarly, in Alzheimer's disease, and some of my research actually comes from cancer research. So it's really important to kind of identify that it's not just one disease, but it's going to be a spectrum, and that we really can kind of help and see things from a global perspective. So what we want to do, start off, is kind of break down what a cell does, particularly what a neuron might be involved in terms of daily function at a cell level, and then kind of just describe some of the therapies and what might happen within the cell itself that can cause a disease like PSP. So this is kind of, I like cartoons, so apologize for this, and this is kind of like the basics of a neuron, and in fact, actually, a neuron is a specialized cell, so all cells will have similar characteristics. Then within the cell, you have this kind of architecture, this formation that Alex has described is supported by the subskeletal elements like microtubules. But within the neurons itself, you'll have different compartments that are really meant to kind of be part of this whole efficiency process. And if you kind of think of things like the nucleus, Golgi, and endoplasmic reticulum, these sound like fairly abstract names, but when you start to distill down what they actually are, the nucleus is sort of like the White House. It's the decision-making part. It's where the genes are. It's where things get processed and produced, and it starts to send out signals. So the endoplasmic reticulum is one of the first things. It kind of surrounds the nucleus, and it's really responsible for identifying what proteins, which are basically the building material of all cells, will get made. This gets transferred over to the Golgi, which is kind of like the processing plant. So it takes that bulk material and starts to fine-tune it a little bit to make sure that things are going smoothly. And finally, you've got the mitochondria. These are the powerhouses of the cells. They're the nuclear plants. They're the ones that produce the energy that are really going to be involved in this whole manufacturing process. So you can see that these things start to work together. And in some of the diseases, these elements might be in conflict with viable neurons. So we know that genetics can play a part in some of the diseases, not just specific mutations, but also factors that might influence the development or increase your odds of developing the disease. We also know that when proteins don't get formed properly, or they're misformed in the Golgi or downstream, that can also become these aggregated forms of tau and synuclein that Alex has talked about. And finally, the mitochondria really has to be active to kind of continue this process. Now, going downstream from the cell body, which is what this is here, we also have basically this network of roads that lead from the cytoskeleton down to the axon, as well as to these dendrites. And this is really important for when you start to think about communication. The cell communicates within itself, but it also needs to communicate with other cells. So Catherine Lee Scott actually talked to me yesterday about some differences where you can start singing a song, and it kind of create some movement, and she was talking about her husband and how to motivate sometimes for movement. And the rationale for this is that some of the memory that one encodes also involves movement. So these two pieces where you have memory and movement together might be really involved in each other. So having this cytoskeletal network and this communication is really essential to keep functioning. And it's also going to be a major part of the therapeutic strategy that we have towards identifying drugs that might benefit and support this whole cytoskeletal communication network. It's really meant to be both form and function, so the form by how the neurons look, as well as function in terms of how it communicates with other neurons and within itself. So what we know from the proteins that are involved with PSP and CBD is that those proteins are called tau, the tau protein. And as Alex has said, it's really part of this whole cytoskeletal. You can see they're here. They're just basically bound to the microtubule network. And then alpha-synuclein, which is involved in MSA. And this is actually part of the communication network. So these are part of these synaptic vesicles that we can see. So this is the kind of the function and where some of these proteins might reside. And why I lump these two together, Alex has mentioned that neurodegenerative diseases are really proteinopathies. So these are proteins that start to kind of create clumps and aggregation, and you can actually see from some of the processes that this is actually where we stand in terms of our understanding. So tau is bound to these microtubules. This is where the cytoskeletal or the form of these neurons that are shown in previous cartoons. And what happens in terms of pathology that we see in the brains of not just PSP and CBD, but also Alzheimer's and on cases Parkinson's disease, is that you have something called phosphorylation. And what happens is the tau protein gets decorated by these phosphate groups, and it's by an enzyme. Some of the phosphate groups are meant to be normal. Others are involved with promotion of their aggregation within the cell. And finally, creating these neurofibrillary tangles that really are hallmarks of Alzheimer's disease and PSP. And when you have these neurofibrillary tangles form, they start to aggregate and lock up the whole cytoskeletal pathway. So, and finally, the other part of the mechanism of what happens in cells is that you can see that when if the cell itself cannot take care of it, or if it happens to start to die off, it can also release that into the environment. So going from one cell to another. And this is kind of the hypothesis that's been going around, or the theory that not only does the aggregated forms of tau affect the cell that it starts in, but it also can start to seed pathology in other neurons. And so this also becomes an important aspect in terms of how the disease progresses over time. It doesn't necessarily happen just within the cell, but it can start to spread from one cell to another. And similarly, synuclein has the same profile in terms of it starts off as a single molecule, and then it starts to aggregate within the cell and causes these fibrils and ultimately Lewy bodies, which are classical hallmarks of Parkinson's disease, but also have been identified in robust cases of MSA. And as with tau, it also tends to spread into other areas. And this kind of recurring theme occurs in other neurodegenerative diseases. So what we try to do is target these proteins in several ways so that we can make sure that we target them at different steps to see which might be the best. And there's actually quite a bit of research that's going on in terms of targeting and identifying what the best place to do that is. Now, before I kind of go into some of the therapeutic strategies, a lot of this is also supplemented by work that's being done in terms of biomarkers. So one of the things that's really come out in the forefront in the last few years is kind of imaging for the suspect proteins of interest. and identifying them. This will start to kind of play into how one diagnoses is earlier, the PSP versus CBD, which often can be having mixed diagnosis. If you can identify the protein tau or synuclein, as well as where the brain region is affected, you can start to kind of identify what diseases might take place. And this can also be supplemented by biomarkers like CSF. And genetics is actually going to be a very important aspect of this. And all of these are programs that CurePSP has funded, as well as our active programs at Columbia University. So in terms of protein target, or in terms of targets for treatment, one of the things that we want to do is just basically look at this aggregated protein and dissect it at different parts to see if we can stop the aggregation and the spread. I'm going to quickly go through some of the pathways and get to the one that I'm working on. And so these are kind of the laundry list of things that are actively taking place. And the first one is to kind of improve the transport. So as I said, transport's really essential. And the reason why is you basically want to bring things moving back and forth in a healthy fashion. And this is also going to be important for when I talk about some of the research to get rid of this garbage. The other one, as I said, is that these phosphate groups get decorated onto the tau protein and starts to initiate the aggregation. So there is a lot of studies that are going on. Lithium is one of the examples of kinase inhibitors. And these are meant to basically stop the initial formation of these aggregates at a very early stage. On the other side of kinases that start to decorate these tau molecules are phosphatases. And if you can enhance these phosphatases, basically they remove these phosphate groups that are aberrant and will start to improve the tau function. Now, another way of actually taking a look at this is we know that the tau molecules and synuclein molecules tend to have a linear form, and when they get phosphorylated, they start to aggregate together. And chaperones are really what it sounds like. The chaperone just takes one person and takes it away from the group. And they're meant to be side by side, let me aid you in terms of this transport. And it helps to restore the function or says, we'll have to kind of recycle this protein for this. And the chaperones are really critical for this whole interplay that goes on within the cell in terms of things that can be restored for function or things that will have to be removed for clearance. And then finally, as I said, a lot of the tau protein and synuclein also gets out of the cell and can start to spread to other cells. This transmission is an important step in terms of enhancing the pathology that occurs. And so immunotherapy has actually been something that's been worked up in other diseases like cancer and Alzheimer's disease. And we're getting to the forefront where we have identified immunotherapies that are targeting the tau protein, specifically the abnormal form of tau, as well as synuclein. So the goal here is to, maybe you won't be able to rescue that first set of cells, but you'll start to be able to rescue the other cells and prevent this kind of downstream, rolling down the hill cascade of neurons that are going to be affected by these general classes of what we call proteinopathies. And so the final kind of research strategy, which is really a translational strategy for drug development, is how do we clear these proteins within the cells to restore function? And this is kind of some of the research that I'm working on, which is involved in kind of identifying small molecules that will enhance autophagic, which is the eating process or proteasomal activity. So going back to the original cartoon, you've got all this functional activity that's going on. And occasionally you'll have some of these proteins start to aggregate out. And what you actually need are kind of this ramp up the clearance of these proteins. And I studied something called autophagy, which is Greek for self-eating. And if you kind of think of, does anyone remember Pac-Man? The video game Pac-Man is really what an autophagic vacuole is. Basically, it's chewing up the garbage as it goes along the path. And what it is this kind of novel like synthesis that goes on within the cell. There's cues that basically say, hey, we need to get rid of this. And it could be the misfolded protein or the aggregated proteins, or potentially lipids, which are associated with Lewy bodies and synucleinopathy. And what it does is it basically envelopes the whole area and wants it to deliver it to the lysosome. And the lysosome is sort of like your stomach. It's filled with acids to basically break down proteins, lipids, and complex carbohydrates. So you end up with amino acids, fatty acids, and simple sugars. And those are really important for your energy, for your cell reconstruction. And this is why the neuron typically is a very efficient process, because this is a normal activity that should go on constantly. And so what we want to do in terms of the therapies is to either promote the creation of these Pac-Man autophagic vacuoles, or increase the movement and association with lysosomes, so you ultimately break down all the material and recycle it. And so when you kind of think of it, I describe the cytoskeleton as this roadway or highway. The autophagic vacuoles are really like the garbage trucks that are taking in all the material and then basically delivering it to the recycling center, which is the lysosome. And that's going to start to clear away all the garbage. And so that's the goal, is to kind of have this. And you can also think of therapies like improving transport, where that can also help some of these activities. There's another pathway that helps to clear out proteins, and that's the proteasome system. A colleague of mine, Dr. Karen Duff, and her postdoc, Mitochondria, recently published on how the proteasome can also be activated to help promote the clearance of these tau proteins. And it could also be applied to synuclein, because we know that the proteasome also breaks it down. And what happens is chaperones that I previously mentioned start to unfold these aggregated proteins, taking them away. And if they decide that they can't be recycled, what they do is they'll deliver to the proteasome. And the proteasome basically is sort of like a paper shredder. You put in one piece of paper and it'll shred out and give you little strips, and those are the amino acids that come out of it. Now, if you take too many shreds, pieces of paper, it'll block it up. And so what we want to do is have a fine-tuned system where you can have multiple proteasomes kind of clearing away the single pieces of paper before you have too many pieces of paper, or these aggregates start to form up within the cell. So these are really represent the two pathways that we can do to kind of remove proteins within the system. And they often work in concert. So this is a cartoon that we often use in the scientific discussions to show that you have a tau protein, and basically things can be decided as to how it's going to be cleared. You can either use the Pac-Man method if it's too large, or you can actually take the chaperones and start to peel them off so they can feed through the proteasome and shred out and remove them. And a lot of strategies that my lab's involved in is to take a look at some of these proteins and see what is the best fit in terms of identifying how you're going to promote these activities to help clear the proteins. And the important thing is, so my research is focused on quite a few diseases: Alzheimer's, Parkinson's, as well as PSP and CBD. Focused on the tau part of this and the synuclein part to understand how we can utilize this pathway for not just PSP or not just Alzheimer's, but really all of these diseases together. And to supplement some of this research, we also have a lot of genetics that's going on. And this is going to start to play into how we strategize for these. Because sometimes the genetics can also affect how we start to clear out some of these proteins. And so understanding these strategies are really important. Often, Alex has talked about using mouse models. We've started to, and he's also worked on stem cells, we've started to kind of explore what stem cells can do in terms of identifying what happens in patient cells versus normal cells. And down the stream, there's actually been discussions about how stem cells might restore some of the function within that. Another funded group actually for CurePSP is screening for new drugs, and sometimes using some of these pathways where we accentuate autophagy or the proteasome system or chaperones that promote transport are really going to take place. And so these are supplemental programs that are really feeding into the whole basic research that is for identification of new drugs and therapies. So to summarize, there are several strategies we can take to approach how to block the protein accumulation. The next part is also that some of these strategies, and some of them actually are already approaching clinical trials. We hope that some of these strategies will start to be able to not only identify what are feasible therapeutic targets, but also what potentially you can take into the clinic. So one of the things that is important in my research, for example, is to make sure that anything that we start to produce can get into the brain. The brain is also very well protected. So you want to make sure that any drug that works isn't working just in the body, but actually gets into the brain, because the brain is typically protected by something called the blood-brain barrier. And that's where all this material that's outside of the brain, there's very little that gets into the brain, because the brain has its own little environment, and it needs to be protected from the outside, because things from the outside can be deleterious or harmful to the brain itself. So identifying these things are really important in terms of the drug discovery part. As I've said repeatedly, is we start to learn a lot from other fields, from Alzheimer's and Parkinson's, because the proteinopathies that occur there are the same as the proteinopathies that occur in PSP, CBD, and MSA. And finally, because of the complexity of the diseases, and while I've simplified it a lot, there's so many brain cells, and there's so many different pathways that can cause the disease. We really need to learn more about how the healthy brain functions, and what's the difference between a healthy brain cell versus one that starts to accumulate these proteins. This really requires the funding and a voice from the community to promote for funding and get governments to contribute to funding. One of the things that I'm extensively involved is advocacy, where we actually are approaching foundations as well as governmental organizations like the federal government to really focus on how we can improve the quality of life and how we can identify therapies for the community. It's very, in a lab it can be a very sterile environment, but I think ultimately we all recognize the importance of why we're doing this research. It's for the patients, it's for the families, it's for the quality of life, it's for a healthy brain. So one of the things that I'm involved with is brain awareness to the public, as well as advocacy to governments. And I encourage you, to express to politicians that this is really something that's important. One of my tasks next week is actually to visit with our local politicians actually in Congress, to encourage them to increase funding for diseases like PSP, CBD, and Alzheimer's and Parkinson's. So hopefully we have your support in this, and if there are any questions, both Alex and I can answer them in the question and answer period that's going to take place right now. So thank you very much.